Okay, welcome. Good morning. Welcome back to FirstThought Focus. FirstThought Focus is our virtual conference series that aims to showcase the breadth and depth of FirstThought's experts within the life sciences industry, while also covering a wide range of topics that are relevant and important to this space. Most recently, we hosted a discussion on AAV vectors in gene therapy with Dr. Gao from the University of Massachusetts Medical School. And you can find the replay of that event as well as others on our website, firstthought.io/focus. Today, we could not be more thrilled to welcome our guest, Dr. Drew Pardoll from Johns Hopkins University School of Medicine, who will be covering with Neil a variety of pertinent topics in the cancer immunotherapy space. And finally, just as a quick reminder before we get started, we will be taking questions from the audience. Really, you can submit these questions at any time throughout the conversation today, and you can submit them via email to hello@firstthought.io. And with that, I'd like to turn things over to our moderator and veteran science journalist, Neil Canavan.
Hi, welcome to First Thought. My name is Neil Canavan, the Managing Director of the KOL Network here at LifeSci, and author of A Cure Within, Scientists Unleashing the Immune System to Kill Cancer. I mention this not only to be self-serving, which certainly is, but also as to serve as partial introduction for my guest today, and that is Dr. Drew Pardoll. Dr. Pardoll is the Professor of Oncology as well as Director of the Bloomberg Kimmel Institute for Cancer Immunotherapy at Johns Hopkins. Further, and how I came to know him, Dr. Pardoll is the subject of Chapter 8 in my book. The reason for his prominence in that book is he is truly one of the pioneers of cancer immunotherapy, having made numerous foundational discoveries in the field. They range from the development of cancer vaccines, the characterization of gamma delta T cells, algorithms for neoantigen prediction, the clinical relevance of what's being called a MSI high patient, and many more. Business-wise, Dr. Pardoll is one of the co-founders of Jounce Therapeutics, a board member of Drayson Pharmaceuticals, a venture partner in Cantelio Capital, the SAB chair of Compugen, on and on and on. The point being, Drew knows this space front and back. So in light of that, as Morgan mentioned, today we're going to be taking Q&A starting now. And I'll fold in your questions as time and topic allows. So enough for introductions and instructions. Let's get started. Drew, how the hell are you? It's good to see you.
Great to see you, Neil, and thanks so much for the invitation. For the audience, Neil and I go way back. Neil was really the first and is in many ways the father within the area of journalism to really take IO immunotherapy seriously as a modality that has the possibility to transform cancer treatment, and really was in the front row in the majority of our key conferences from literally, I can't remember how many years back at this point. And so other than that Chapter 8, I think Neil's book is a great read, and it does actually capture through individuals in the field a perspective that I really don't think any other writer has captured.
Wow, thank you. I mean, there'll be many more questions about me later. But first, we're going to dive right in with something called TIGIT. Drew, 5 points. What does TIGIT stand for?
Boy, you stumped me right out of
It's a trick question. What they call it is T cell immunoglobulin and ITIM domain. However, I spoke with Jane Groden, who is accredited with the discovery of TIGIT, and she assures me TIG, the first letters, are the initials for her daughter. And I asked Mike Callis about this because they work together, and he goes, yeah, that's the story he told me. She told me to. So anyway, that aside, can we have the first graphic, please?
See, I've already lost my credibility.
There we go. Okay, TIGIT. Drew, why should anyone care about TIGIT? Here we are.
Right. So TIGIT is a checkpoint in that its engagement largely with CD155, which is also called PVR. It's actually the poliovirus receptor, will inhibit T cell function. It falls into this group of checkpoints, of which PD-1 is arguably sort of the father, but also LAG3 and TIM3 fall into this category of being upregulated when a T cell is activated, and it essentially represents part of this feedback inhibition system that all components of the immune system has to regulate to essentially fine tune the amplitude and character of their response. TIGIT was, there were actually a few groups that discovered TIGIT simultaneously. Genentech was one. And it is part of a fairly complicated set or family of regulators, such that what's actually shown on this slide is actually highly incomplete. As I'm guessing that this was put together probably, this particular figure is a number of years old.
I'm guessing about 2 years old, yeah.
Yeah. It has some similarities to the original B7 CD28 interacting with B7 also interacting with CTLA4. And you see CD226 on there. There are other members of the TIGIT family that look like they are parallel inhibitors, one of which is called PVRIG. And then there's another receptor called CD96. And I think this brings up some of the challenges of blocking these checkpoints, is that most of them are not simply a one-on-one. You have one ligand or partner and then one inhibitory receptor. You block that, and that's the whole story. In a lot of these cases, there are multiple parallel family members, and cases where you can have interactions that are activating as opposed to inhibitory, and figuring out how to navigate that complex biological wiring into a particular therapeutic agent that enhances an anti-tumor response is not trivial.
I want to drill down a bit more on that in a few minutes with the PVIRG story. But first, to go back, the reason I chose that sketch is because it was simpler. I mean, as we both know, this is a horribly complex topic, these checkpoints. And just a quick aside to the audience, this is a checkpoint t-shirt from the band. The one Drew has on is the original shirt. So we are honoring the band today and checkpoints. So back to TIGIT, I ran across an interview recently by Vinjay Kutru, and he was talking about this mouse model he made where he knocked out TIGIT only on B cells. This led to multi-organ autoimmunity. The B cells were not in that graphic. In the context of cancer, is TIGIT and B cells relevant?
completely not known. The B cell specific knockout story of VJs is really interesting, and it really came out of left field. It would have been nothing that would have been predicted. TIGIT is also expressed on NK cells. And that's also an additional layer of complexity, which is the different cellular expression of TIGIT. And yeah, at this point, we don't know what that B cell knockout story ultimately has to do with cancer, and it's still not even 100% clear mechanistically how it's inducing autoimmunity. One of the real controversies with TIGIT now has to do with whether the blocking antibodies to TIGIT, what the constant region or the so-called FC region of the antibody is, and whether it can bind to FC receptors and potentially mediate, rather than just straight blocking of the interaction of TIGIT with PVR or CD155, whether you actually want an antibody that can kill a TIGIT expressing cell. The reason it's conceptually you might want to do it is that the cells that express the highest levels of TIGIT are actually not T cells, not B cells, not conventional killer T cells, but they're regulatory T cells. Oh. So all of a sudden you've got these Tregs, which is this population of general, again, part of this very involved regulatory negative feedback inhibition system, of which there are 100 of these, call them all in this, bucket of checkpoints that such that your immune system, when it's responding to a hepatitis virus, won't knock off your liver in the process. Right. When you're responding to a pneumonia, you don't destroy your lungs. It has to be very, very highly regulated. And so one idea is that you want to have anti-TIGIT antibodies that have an FC that can pull in NK cells and macrophages that have these receptors for the FC region of an antibody, that the antibody will direct them to kill the cell that's expressing the target. And so you can selectively potentially get rid of Treg cells, but at the same time, you've got a window because the anti-tumor killer cells also express TIGIT where it's inhibitory. And so this is one of the controversies in the field, which is, does your anti-TIGIT antibody need to be able to bind to an FC receptor on a cell that can kill target expressing the TIGIT?
Yeah, I mean, okay, so it's already clear to me there's a lot of science that's still to be worked out. Yet, as always, pharma has plowed forward, and there's all sorts of clinical trials. So, while there is some single agent activity with TIGITs, all the programs I'm seeing going forward are with combinations. Merck has their TIGIT asset with Pembro, their anti-PD-1, their anti-PD-1 agent. That's in a trial of lung cancer. Also in lung, Roche is combining their anti-TIGIT asset with anti-PD-L1, Atezo. BMS is doing a basket trial of solid tumors with TIGIT and Nivo, their anti-PD-1 drug. This might be a bit crass to ask, but this choice of anti-PD-1 or anti-PD-L1, does it matter mechanistically, or does this speak more to marketing strategies?
Yeah, so I mean, the reason that Roche Genentech is developing anti-PD-L1 is because they sort of put their chips down on it early, and they're sort of sticking with that horse. I think if you're to ask 100 people in the immuno-oncology world, would you prefer to use an anti-PD-L1 or an anti-PD-1? Actually, I've been on panels where that question was asked and the audience clicks, a thing, and then they tally it up. About 90% would say I'd rather use an anti-PD-1. The other 10% would probably say doesn't really matter. The theoretical reason why an anti-PD-1 antibody might be somewhat better is that there's a second partner or ligand for PD-1, which is now commonly called PD-L2, that most people don't think a lot about. And the anti-PD-1 antibodies block the interaction of both PD-L1 and PD-L2 with PD-1, whereas the anti-PD-L1 antibodies still leave available the interaction between PD-L2 and PD-1, which is not blocked by the anti-PD-L1 antibodies. The general scope of cancers that are so-called responsive to PD-1 pathway blockers is the same between PD-1 and PD-L1. There's been one meta-analysis done looking at 19 different trials and concluding that on that analysis, the anti-PD-1s are somewhat more effective based on things like response rate and overall survival. The reality is that no one has done the head-to-head comparison. Yeah. And in a randomized trial, and for better or for worse, it's probably not going to be done. So I think we're left with sort of meta-analyses that suggest that the activity is similar, but the anti-PD-1s are maybe a little bit more potent.
So with that, of the three combinations mentioned, I'm guessing you prefer the PD-1 combinations, or we have to go back to exactly how the antibody was put together? I mean, do you have a preference of these three combinations?
So essentially, the approved anti-PD-1s are identical. Mm-hmm. Pembro, Nivo, Regeneron's, I'm trying to remember the name, they're all the same. They're in terms of their activity and their side effect profile. So I would probably prefer combinations with, like most people, with anti-PD-1 versus anti-PD-L1. I think all you have to do is look at all of the new antibodies that are being brought in by a gazillion companies that block the PD-1 pathway. And other than the original three, atezolizumab, durvalumab from AZ, and velumab, which was from Merck Serono, that were in that early group, not everyone that's being developed that I'm aware of is anti-PD-1. So I think that sort of reflects a general consensus that those are somewhat better. Now, there's only been one randomized trial between a PD-1 blocker alone versus a PD-1 blocker plus an anti-TIGIT, and that's the Roche Genentech trial in non-small cell lung cancer, where in that trial, their essentially atezolizumab, their anti-PD-L1 plus their anti-TIGIT had a statistically improved progression-free survival, and I believe also overall survival, and actually improved response rate over their anti-PD-L1 alone. So that's the one and only randomized trial that clearly shows that TIGIT added something. However, if you look at their populations, either total or subgrouped by level of PD-L1 expression,
Right.
You have to be very, very careful and take with a big grain of salt a comparison between groups in two different trials as opposed to a randomized trial. But with that major caveat, if you look at the clinical, the response rates, and basically performance in that randomized trial, the anti-PD-L1 plus TIGIT, while being better than the anti-PD-L1, was essentially equivalent to what Merck has reported with anti-PD-1 alone, Pembro alone,
Right.
in PD-L1 high lung cancer. So one could argue, and similar to what BMS has reported as well with nivolumab. So one could argue that Genentech has shown that if you have a slightly inferior PD-1 blocker, you can make it as good, which is anti-PD-L1, you can make it sort of equivalent to an anti-PD-1 if you add the anti-TIGIT. So what's not clear is if you now are at the level of an anti-PD-1, which is a little bit better, are you going to do better still if you add an anti-TIGIT to that?
Okay.
And those are the randomized trials that that Merck is doing, and we'll see where they go. And I guess BMS is also, I don't know if they've started, I think they may have just started a randomized trial. Merck and Genentech are further along.
Well, there's a fourth combination I want to talk about. You've already alluded to it. This cartoon, let me see, this is from graphic number 2. This is from Cancer Discovery of this year, and you co-authored this paper.
Mm-hmm.
So in the lower left, we see the molecule PVIRG, also called CD112R, which along with TIGIT is part of something called the DNAM axis. The Israeli company Compugen is looking to target both these molecules in a combination with Nivo. I realize you're the chair of Compugen's SAB. There's a lot you can't really say, but if you could just please walk us through why this isn't mechanistic overkill.
Right. So, and also in full disclosure, I'm also a co-author on the original paper that demonstrated that PVRIG is also a checkpoint. And so you can, and then there's this third receptor, CD96. DNAM1, so both PVR, which is the major partner for TIGIT, also somewhat for CD96, and PVRL2, which is also called Nectin-2, which is the partner for PVRIG. You can begin to see how complicated this is. They both will also bind to a molecule called DNAM1, which is also called CD226. And that gives you a co-stimulatory signal. So you can see that there is this sort of balance between a co-stimulatory and co-inhibitory signals. That conceptually looks very much like the original story with B7, 1 and 2, that can bind to the co-stimulatory receptor CD28, but you then activate a T cell, CTLA-4 comes up, and now that has a higher affinity, and that will sort of now turn off the CD28 co-stimulatory signal, will dominate. Very similar thing here. The idea is that if you leaving CD96 to the side, and the issue with CD96 is that the literature with CD96 is all over the place. There are some sets of experiments that suggest that it's co-stimulatory. There are some that suggest that it's co-inhibitory. It's really very, very unclear. It is very consistently clear that TIGIT and PVRIG are inhibitory checkpoints on T cells. And by virtue of the fact that their respective partners also bind to DNAM1, and there are other sequence similarities, it's reasonable to put them in sort of in this one family. And the reason we call it the DNAM family is the DNAM is the one molecule that sort of brings together the family, in that the partners PVR and PVRL2, they all bind to and will trigger DNAM. The idea, and in blocking both PVRIG and TIGIT, is that if they're both providing somewhat redundant signals, then blocking only one is going to leave the other available for signaling. I think the example there would be B7-1, B7-2 binding to CTLA. They both bind to CTLA-4. If you block B7-1, you still leave the B7-2 CTLA-4 interaction. If you block B7-2 alone, it's the other way around. There are, I'll just say this from our lab, there are ongoing studies in the mouse that support the notion that you do better if you block both simultaneously. And also, as with almost all of these checkpoint inhibitors, they're all going to end up being used together with a PD-1 pathway blocker, be it an anti-PD-1 or an anti-PD-L1. And the clinical trials that Compugen has started up with the combination of the anti-PD-1, which is nivolumab, anti-TIGIT, and anti-PVRIG as a triplet, are I'm holding my breath to see how they go.
Okay. So overall, regarding TIGIT, do you think this will have the same therapeutic range of IPI or NIVO insofar as checkpoints work best in settings of high TMB, like lung, but not so much ovarian? You think it'll be roughly the same as that?
So I think they will work better when you have a more inflammatory tumor. There tends to be a correlation between TMB, total mutational burden, number of mutations, for those who aren't familiar with the lingo, and immune infiltration, particularly with T cells, which in turn correlates with response to anti-PD-1. And so I would imagine that those are the tumors that blockade of these pathways will work better. And that correlates with what Genentech found in their trial, which is that the benefit of TIGIT plus anti-PD-L1 relative to PD-L1 alone was seen exclusively in the PD-L1 positive tumors. So, so I would say that now, remember, there's TMB is a continuum. PD-L1 expression is a continuum. CD8 infiltration is a continuum. And while on average, if you look at, you know, the 1,000 tumors in TCGA, they tend to track together. For an individual tumor, you can get some real dissociations. You can get tumors with quite low TMB with a reasonable T cell infiltrate and PD-L1 that will be more responsive. But in general, it's that group of tumors that tend to have more mutations, more PD-L1, more infiltration that you're going to do better. The really cold tumors or immune deserts, to use Ira Mellman's term, where you just have no T cells in the tumor, things like you know, pancreas cancer, right, brain cancer. You know, I don't think simply adding TIGIT's going to help.
Question just came in. We can address this one briefly. Do you think that the PD-1 TIGIT PVRG IRG will become the IO backbone of the future? Should I ask you next year?
Ask me next year. I really don't know. I mean, the closest I could say is that if the data shows that it really, really, you know, improves the response relative to PD-1, and it looks like it really adds to, you know, PD-1 plus TIGIT, it certainly could be. But I think it just all remains to be seen. I think what is a concept that I'm glad to see happening is that this artificial barrier in terms of number of checkpoints to block is beginning to dissolve. There's these checkpoints have different biologies. They add, they regulate in different ways. They are not totally redundant. So there shouldn't a priori be a ceiling to the number that one uses. But I think even more importantly than that, there absolutely needs to be more investment in biomarkers to help figure out, you know, does Mr. Jones need all of them, or do they need, you know, just, you know, CTLA-4 and PD-1 blockade? Or does Mrs. Smith, would you do much better if you add anti-LAG3, but don't worry about the TIGIT axis?
Okay, you've anticipated my next topic, LAG3. This has been on your radar since at least 2004. You wrote a paper with Dario Vignali. And according to the cartoon I have, let me see, there it is. LAG3 seems to be everywhere. So is there a brief explanation as why it has taken so long to develop? I mean, there has been some progress recently, but 2004, you were writing about it. Is this a hard target to hit?
So again, it's complicated.
No.
We used to joke that it's called lag for a reason, because the clinical development lagged far behind what we would have liked it to be. So I think there are two issues with lag that have made it not totally straightforward, because none of these are. Firstly, if you go back to our paper where we mated LAG3 knockout mice to PD-1 knockout mice, so they're both they're both knocked out together. PD-1 knockout mice, particularly if they're in a clean facility, don't get any autoimmunity until, they're, a year old, which is about half the lifetime of a mouse. And actually, fortunately for Tsuku Hanjo and the field, he had a dirty mouse facility. So his PD-1, he was the first to knock it out. He discovered PD-1. Those animals in about 9 months began to develop autoimmune phenotypes. In our super clean mouse facility, the PD-1 knockouts never develop an autoimmune phenotype. But the combination, and LAG3 knockouts, no phenotype on their own. You put the two together and the mice look great for 4 to 6 weeks. And then they start dying. And it's really interesting because you'd have a cage, they'd look good, and then you'd come in the next day and one was dead, and the next day another 2 had dropped off. And it turned out that when you combine those 2 with the complete knockout, they die of an autoimmune cardiomyopathy, myocarditis. And so in the original development with BMS, there was a lot of care given to, worrying about this as a toxicity as you went up the dose with anti-LAG3. And so so getting the dosing to the point where there was a comfort zone between added efficacy to PD-1 alone. And we showed in 2012 that really the best anti-tumor efficacy is when you combine them. So that's taken a while. The other thing that's a wrinkle is that it was always thought that the partner for LAG3 was MHC class 2. Actually, it turns out if you go back and look, the antibody that we first used, that actually Dario Vignali made, and that's how that collaboration came about for the 2004, that first paper, that antibody actually does not really block the LAG3 interaction with MHC class 2, but it still has the biological effect. And that may be because MHC class 2 is not the relevant partner for LAG3. It's FGL1, which was only discovered and reported 2.5 years ago by Liping Chen.
Okay.
So we don't know if there are 6, 7, 8 anti-LAG3 antibodies that are in development. And I'm assuming they were all selected to be antibodies that could block the LAG3 interaction with MHC class 2, but we don't, at least I don't know which of those, at least I know for some, but not for others, which ones block the interaction with FGL1, which may be the real interaction that triggers the LAG3 checkpoint. So these are the complexities that have slowed down the development.
Okay, I want to go to one specific asset. This question just came in from an investor, and this is actually anticipated the next graphic I was going to use, which is graphic number 4. If Morgan, if you could put that up for me. Okay, kind of busy, but it shows us where the expression is. And the question that came in is, I'll preface this. This is how it actually came in. My main man, Drew, I am curious to get your thoughts on the BMS LAG3 data from ASCO and how that may be extended to other treatment settings. To remind listeners, the data from ASCO was in melanoma. there was a PFS of 10.1 for the combination versus 4.6 for Nivo alone. So Drew, given the expression profile on this table, can we move on to other tumor types?
Yeah, so there is a lot of variability from tumor to tumor, tumor type to tumor type in LAG3 expression. Again, when you tend to look at populations of tumors, there is a general correlation with PD-1 and PD-L1. But when you start looking at individual cancers, you can see a lot of variability. There are certain cancers, so for example, in Hodgkin's, particularly EBV positive Hodgkin lymphoma, LAG3 is really, really high. I don't know if nasopharyngeal is on there, but nasopharyngeal is very high. A lot of the virus-associated cancers, it's pretty high. Some of these other guys, I wouldn't say it's necessarily high in every tumor type. So it's very variable, and I think we need to do a lot better in defining how LAG3 expression patterns will predict and guide the use of anti-LAG3 antibodies on a patient per patient level, so-called what I call precision immunotherapy. What BMS did was just very, very top line. It was essentially the number of LAG3 positive cells relative to all nucleated cells. In other words, relative to all cells. Mm-hmm. So if you had very few T cells in there, they could all be LAG3 positive and you would still get a low LAG3 score. So what they didn't do is to look at LAG3 expression on particular subsets of T cells in the tumor. They didn't even look to see, so if it's 1% LAG3, that's 1% of total nucleated cells. But is that because there were relatively few T cells, but all the T cells were LAG3 positive, or were there lots of T cells and it was a small proportion of the T cells that were LAG3 positive? So it's going to require, and certainly we and other groups are putting in a lot of effort into getting a much more refined, higher dimensional pathology, either immunofluorescence or multidimensional immunohistochemistry to better refine that.
Okay, I want to move on to one more. If I could have graphic number 5. This is sort of a segue between LAG3 and something else that we've tried to talk about over the years. And let me get my graphic up, just a moment. Okay, well, the graphic will be up in a minute, but what it shows is something called TIM3. Now, I had hoped that TIM3 was actually named after a person like TIGIT, but it's not. It stands for T cell immunoglobulin and mucin domain containing 3, or TIM3. I used to hear a lot about it. Now I don't. And this question came in just now from an investor. LAG3 was revived. So how should we think about other exhaustion-related targets like TIM3? Is there a path forward here?
Right. So LAG3 was not revived because LAG3 never died. What happened was it basically went dark for the phase 3 trial. And, this often happens in oncology, is you have various phase 1, phase 2 trials, And then at some point or other, a randomized phase 3 trial is done, and there's nothing reported until there's the final report. If you actually followed it on clinicaltrials.gov, the LAG-3 Nivo trial, there was a randomized phase 2 component, and then there was a pause for about 6 months. And then it all of a sudden there was accrual again, and then there was the announcement. So just to sort of put a fine point on this notion of, if you don't hear something about a particular agent, does that mean it's dead, or does that mean it's being studied clinically, but it's just gone dark? I think that's part of it with TIM3, is I think some of it is that after the IDO debacle with melanoma, where there was a lot of hoopla, and then they did the randomized trial and the two curves completely overlapped. That sort of created this, I think, sense in the oncology world, including the investment community, that, these single arm, 40 patients, 50 patient phase 2 trials are interesting, but show me the clean randomized trial with enough patients to give you a good P. So what I thought, I could be wrong, but was that some of the TIM3s are now in this dark period where, together with anti-PD-1, there are some randomized phase 3 trials. That may be why it may be the same phenomenon, and we may hear in a year or two that, there's something really interesting. TIM3 is a checkpoint. When you knock it out, you can clearly see that it's a checkpoint, and the cleanest way to define something as a checkpoint is to knock out the gene and then look at what happens in the immune response, anti-tumor or otherwise. It is definitely a checkpoint. The big challenge with TIM3 is that there have been 4 or 5 partners or ligands that have been reported for TIM3. There's phosphatidylserine, there's, which I forget which one of the galectins, one of the high mobility group, HMGB1, I think, has been reported, and there are 1 or 2 others. So the problem is when you have, and it's not clear that these aren't all real partners, although in some cases they could be interactions that you can define biochemically, but they're not biologically relevant. The problem when you have that is that if you don't know what the biologically relevant partner is, it becomes hard to know whether your particular antibody is actually blocking that interaction, which is biologically relevant. So was the same thing with LAG-3. I will say that, we developed what's essentially the precursor to relatlimab from BMS with Medarex, and we actually used a functional assay. So we didn't only just say, okay, whatever blocks the binding to class 2, because we knew that that wasn't necessarily the biologically relevant interaction. So we said, let's make sure that our antibody does something functionally. Though I think that's been one of the challenges with TIM3, where there are many reported partners, and figuring out whether your particular TIM3 antibody is blocking the relevant interaction. So commonly, it's shown as binding to as the partner is galectin-9, But again, that's an oversimplification. You've got other, I mean, I can tell you there's some people that feel very strongly that phosphatidylserine is the relevant interactor with TIM3. But there are others as well.
All right, we have 10 minutes remaining. There are some questions coming in. I'll remind investors on the call, get your questions in now. We have about 10 minutes left. This one is completely different from what we've been talking about. This is cell therapy. What was the question? About gamma delta T cell. Oh, all right. Do gamma delta T cells have sufficient potency as anti-cancer agents? And a follow-up question is, do you have an opinion on gamma delta T cell engagers? And for the audience's knowledge, gamma delta, Drew is one of the foundational discoverers of this cell type.
Appreciate that shout out. So this is what I'll say. When triggered in the right way, gamma delta T cells can kill as potently as any CD8 cytotoxic T cell. So they definitely have the weaponry. The big challenge with gamma delta T cells is that we still have surprisingly little knowledge. I should say I haven't worked directly on gamma delta T cells for a while, but there's still a lot of question marks as to what their gamma delta receptor is recognizing that's relevant in the tumor. And you typically, in most tumors, you don't see a lot of gamma delta T cells that are naturally there, in part because gamma delta T cells are largely part of the mucosal barrier immune response. So you find a lot of them in the gut, actually sort of intercalated in the gut epithelium. You find them in the bronchus. You basically find them in epithelial linings. You find them in the urethra, in the vagina. So it's maybe not that surprising that you don't see a lot of them in your typical tumors. The question is, if you artificially, you develop an engager that will, where one arm is against an antigen expressed highly by the tumor cell, and the other arm is an antibody that can trigger gamma delta cells, can you focus their killing activity into the tumor? Mm-hmm. And I think it's still early. I think if you can, they have the potential to be very potent killers. Is it something that if I had one bet to put on that I would bet on? I would say no. Oh. Relative to the other stuff that's going on, I certainly hope that they do add something on behalf of cancer patients. And, as one of the people who sort of simultaneously stumbled onto them many years ago, it would be great to see them doing something therapeutic. But right now, I think it's very early, and I think there's just a lot of other stuff that's further along that I think looks promising.
This is a follow-up to that question, and this invokes the spirit of Dr. Steinman. When thinking about other cell types, particularly myeloid cells and dendritic cells, what are targets that are really interesting and that have clinical trials on the horizon?
Myeloid cells incorporate multiple different cell types that include dendritic cells, macrophages, and these cells that are commonly referred to as myeloid-derived suppressor cells, or MDSCs. And I think they are all important. There are some very interesting, so one of the things that we're beginning to appreciate is that everybody thought of dendritic cells as being the critical cell type for vaccination, which they are. But people really still don't think of dendritic cells as being important players in, for example, checkpoint blockade. Turns out they are.
Yeah.
In fact, in this whole neoadjuvant PD-1 story, for example, it looks like one of the important roles of PD-1, and this was actually when we defined PDL2, we put a lot of emphasis on this, is modifying the initial activation of T cells by dendritic cells expressing antigen. In fact, when we cloned PDL2, we called it B7DC because a B7 family member expressed highly on dendritic cells, which it is, in addition to being a PD-1 ligand. So there are some very interesting molecules, some of which I can't name because they haven't been published yet, but that are selectively expressed on dendritic cells. And there are others actually that have been reported, like the CD200 ligand pair that will block the ability of dendritic cells to stimulate T cells. So there are some very interesting targets on DCs that are, I think, going to turn out to be very promising, completely apart from cancer vaccines. Then, macrophages and myeloid-derived suppressor cells, there are a number of targets. Obviously, there's a lot of interest in CD47 and also CD24, these so-called don't eat me signals, but there are many other interesting targets. In not enough time to go through them all, but there are. And some of the most interesting targets are metabolic targets. And this is a whole area unto itself, which is metabolism. Everything that an immune cell does.
Drew, let me stop you because I'm going to have Jonathan Powell on. And actually, I was going to ask you about metabolism, but we're running out of time. I have one question that came in from an investor. If we could deal with that shortly. And then right after that, I want you to tell me about MANA. Okay? Because you mentioned cancer vaccines. So the question from the investor, what checkpoint pathways appears to be most important in autoimmunity? Short answer.
Oh, clearly the two most significant ones are CTLA-4 and PD-1.
Okay, short answer.